the digestive system. So for this system, we're going to look at all the organs and we're going to outline every single structure starting at the mouth and working all the way down to our anus. So this picture does pretty good, or gives us a pretty good representation of some of the major structures, some of the major organs that are involved in the digestive system. We can start at the top looking at our mouth, which is going to contain our teeth, our tongue, that's going to be used for certain processes of chewing, otherwise known as mastication. We'll look at the glands in the mouth that are involved in moistening, starting enzymatic degradation or breakdown of this food product. The sublingual gland, the parotid gland, the submandibular gland. Then we have that pharynx, that funnel-shaped tube that we talked about in respiratory system. Again, here the oropharynx, that passageway from the mouth down into the esophagus. The esophagus is going to tie up and meet with our stomach, then our small intestine, the portions thereof, the large intestine, the sigmoid colon, the rectum, etc., etc., etc. So, we will look at some accessory organs like the liver, the gallbladder, the pancreas, and its part in digestion. We will also look at the layers of protection we have with the peritoneum and all the different folds that surround and encase, provide blood, nutrients, nerve stimulation, and lymphatics to these structures. Remember, this is one tube that starts at the mouth, moves to the anus, and basically just changes shape, function, and size as it moves down. It is open to the surface, which is why it is going to be lined with epithelium. When we did epithelium in AP1, the majority of these organs were covered because this is one continuous open alimentary canal. So as a review, we can look at our gastrointestinal tract. This GI tube is long. It's open at both ends. That's going to allow for the transit of food during processing. Some of the portions of this tube includes the esophagus, stomach, 3 portions of the small intestine, the large intestine, and the rectum. We do have some accessory structures that assist in the breakdown and processing of food. These accessory structures include our teeth, our tongue, our salivary glands, liver, gallbladder, and pancreas. When we look at the overall process of digestion, we can break it down into 6 basic processes. The first being ingestion. So food is going to move into the mouth. We're going to get a series of secretions. So glands that are going to secrete digestive enzymes. That's going to help break down the products of this food. We get motility, so mixing and propulsion of food and secretions. We talk about wave-like peristaltic contractions and circular contractions. We deal with digestion. So we go from large food particles, we use those digestive enzymes to break them down into smaller food molecules. So for example, if we start with a polysaccharide, our goal is to get it into a monosaccharide, a simple sugar. So we can work our way from large components breaking down into the smallest components so that it's easily digestible. So that's digestion. Second is going to be absorption. So we're going to be absorbing those nutrients into the blood or lymphatic vessels. And lastly, we have defecation. So removing any waste, byproducts, bits of food that we could not digest or absorb, we're going to eliminate through this process of defecation out the anus. The layers of the gastrointestinal tract. Now, as we examine this tube as it moves from mouth to anus, we have to examine some of the layers that are making up this tube and separating them so that nerves can come through, so that muscles can contribute to action, so that glands can be embedded and parts of lymphatics. So we start at the space, the lumen. So we look right here. This is going to be our lumen. This is the space with which that food or digested material is going to be moving through. We look at that epithelial line. So we outline this epithelial line right here. So let's zoom on in. We outline this epithelial line, and we'll see that we're going to call our epithelial line and the tissue the mucosa. Now the mucosa is going to incorporate that epithelial line, this lamina propria below it, and a layer of muscle. That layer of muscle is going to be called the muscularis mucosae. The main function of this portion of the mucosa is going to be to aid in propelling nutrients in one direction, from the lumen to the submucosa. Now, while we are still in this area, we need to look at a few other things. The first are going to be these little green patches. This is our mucosa-associated lymphatic tissue. We talked about this when we did our lymphatic lecture. We used our Peyer's patches as an example. Remember, we said that it was found in the small intestine and the ileum. We're also going to zoom out. We're going to find that we have some mucosal glands. So here's one, here's another one, and here's another one. The main function of these glands in conjunction with the epithelium is going to be to aid in absorption and secretion. And the secretion is going to be of mucus. The next layer we're going to look at is going to be the submucosa. Now, as its name implies, it is going to be directly below the mucosa. It's going to support blood vessels, it's going to have some glands in there, and it's going to have a submucosal plexus. First, the submucosa is going to be comprised of a layer of dense, irregular connective tissue, or even loose connective tissue. Now, the glands that are located within, so we have submucosal glands, are going to vary. In the esophagus, we'll have esophageal glands. Again, these are going to be secreting mucus onto the surface to facilitate movement through the esophagus. In the small intestine, as an example, we have Brunner's glands, again, mucus-secreting glands. And then we see this submucosal plexus. This plexus is going to function to regulate the configuration of the luminal surface, so controls contractions as well as glandular secretions, and it's going to alter electrolyte and water transport. It's also going to play a major role. Now look right here. See, this blood vessel is coming into this layer in this area, and we said that we are supporting blood vessels. You can see that artery and vein there, the artery and vein here, and that nerve here. It's also going to regulate local blood flow. The next and third layer is known as the muscularis. So the muscularis is responsible for those segmental contractions and peristaltic movements within the gastrointestinal tract. We find two layers. Now, we have an exception to this, and that's going to be in the stomach, where we find a third layer. But those two layers are smooth muscles that are going to be arranged in an inner circular layer and a longitudinal outer muscular layer. Now, again, in the stomach, because that stomach is going to function as a bulk churning chamber, we put in a third layer, an oblique layer that gives us pretty much coverage on all three planes, allowing for max churning and twisting. Think of a washing machine on spin cycle. These three layers of muscle is going to allow us to accomplish that. So we could zoom on out. We'll get back into a larger view. And we find one other structure here. It is the myenteric plexus. So this is our second nerve plexus. Now, this nerve plexus goes to these 2 or 3 layers of muscles, and its primary responsibility is going to be those peristaltic movements of the bowels. Now, it can act independently from the central nervous system. It receives innervation mostly from the autonomic nervous system. So we go back to that fight or flight response. When we're talking about fight or flight for our sympathetic and parasympathetic rest and digest, this is the nerve plexus that is controlling that, and ultimately connecting the central and enteric nervous systems to the autonomic nervous system. The fourth and final layer that we're going to look at is going to be the serosa. So we see it down here. It's going to be comprised of areolar connective tissue and a layer of epithelium. The serosa is a smooth membrane. It's going to consist of a thin layer of cells found on the outer wall of the organs of the abdominal cavity. And that abdominal cavity is the serous cavity. We've talked about this. And we can look a little bit further and we find the mesentery up here. We've talked about serous membranes. Now, anywhere along this alimentary canal, from mouth to anus, anywhere there is a peritoneum, so a serous membrane, the outermost layer is going to be lined with the serosa. Now, anywhere that this tube lies outside of this abdominal or serous cavity, it will not be lined by a serosa, but rather a layer called the adventitia. Now we can outline the specific parts of this tube that is surrounded by this adventitia that is not outlined by a serous membrane. And this includes the oral cavity, the thoracic esophagus, the ascending colon, the descending colon, and the rectum. It is these structures that have that adventitia that we outlined here instead of the serosa. Neural innervation of the gut. We've outlined this before with the picture on the previous slide. We've talked about the enteric nervous system working with the submucosal plexus, dealing with the secretions from the glands in the submucosa, whether it be in the esophagus, whether it be Brunner's glands in the duodenum, the first portion of the small intestine. We also highlighted the myenteric plexus, right here, the myenteric plexus, and its job in regulating peristaltic contractions. Now, we talked about the autonomic nervous system where parasympathetic innervation is going to increase motility and mobility through the GI tract through muscular contraction and control of these 2 layers of muscles, or 3 if you're in the stomach, when we are resting and digesting. And the sympathetic will tend to suppress movement of the digestive tract and the organs of the digestive system when we are in a fight or flight or stress scenario. We can examine the GI reflex pathways looking at sensory information coming in via our sensory neurons and our motor response out. So if we start at the mucosal epithelium, we have sensory neurons that are detecting change, food entering this pathway, and we see that we innervate to the submucosal plexus, so we come up to the submucosal plexus, and we also ascend to the myenteric plexus and to the autonomic nervous system and the central nervous system, at which point that sensory signal will be interpreted and a motor response will be delegated. So we have here our motor response traveling down to those longitudinal and circular smooth muscle layers within that muscularis, initiating those wave-like peristaltic contractions. We have some interneurons here, so if we go to a purple, well, we'll keep it green. We have an interneuron that is mitigating that information, that is interpreting that sensory information, that is communicating with the myenteric plexus as well as the submucosal plexus. Now, the information, the motor response from the submucosal plexus is talking to the submucosal epithelium, allowing those glands to secrete their substances onto the surface of the lumen. That brings us to the peritoneum, the serous membrane that protects all these organs within our abdominal cavity. Now note that the peritoneum is the largest serous membrane that we have in the body. And we break it down into portions, and their name in some instances will tell us where they are located. So first, we're going to look at the stomach. It is located right here. And we're going to zoom in. I'm going to do my best to draw a nice stomach. So it is so here's our esophagus coming in. It is a bulbous J-shaped structure. Okay, that is a pretty bad stomach. So we have a sphincter up here. We have a sphincter down here. Now, when we talk about the stomach, we deal with a couple curvatures. We have a greater curvature, and a lesser curvature. And we are going to look at this again when we look at the anatomy of the stomach, but why is this important? Why am I taking the time to outline this? Well, if we look at the peritoneum, we see that we have a greater omentum and a lesser omentum. The lesser omentum is going to originate from right here in that lesser curvature, and it's going to extend up and behind the liver. The greater omentum originates from here, the greater curvature of our stomach, and it's going to flap down over the entire abdominal cavity. It is essentially like an apron. It is this big fatty apron that serves a function of energy stores, cushion, protection for all the organs lying behind there. Remember, when we talked AP1, we talked about the abdominal cavity, how it's a unique cavity in that there isn't much bony protection here. Not like the thoracic cavity that has the sternum, the ribs, right? Not like the pelvic cavity that has those 2 huge os coxae, or the brain, the cranial cavity that has those cranial and facial bones. So pretty unique that we have to protect it with this peritoneum, which is why it is the largest serous membrane in the body. Now, the next one we're going to see is right here, the mesentery. So we circle it here, and we see that mesentery is going to move between all the portions of the small intestine. So its job essentially is to support the small intestine, and we could follow it through all the different regions here, right? And we're going to provide blood, nutrients, oxygen, nerve supply, and lymphatics to all three regions of the small intestine. That brings us to our mesocolon. Now remember, we said that the ascending colon, the descending colon was not surrounded by a serous membrane. Instead, it was surrounded by an adventitia. Well, we do have a serous membrane that surrounds portion of the colon, and the portion that it surrounds is the transverse colon. So we will write transverse colon here. Okay, so it is the only part of the colon that is surrounded by a peritoneum, by a serous membrane, by a serosa, and it is called the mesocolon. Now, if we go back to our arrangement of this serous membrane, remember the innermost layer is called the visceral peritoneum, and the outermost layer is called the parietal peritoneum. So we can continue on with the peritoneum and change our view a little bit. Instead of a mid-sagittal, look at a nice frontal view because it gives us a good look at some of these folds. So we start with the greater omentum, so we see it right here. And again, I'm not a big fan of this because this picture shows it originating or attached to the large intestine. Remember that this greater omentum originates from here, from the greater curvature of the stomach, and will hang down like this big fatty apron over the abdominal organs. We can also see, we can lift the liver up on this picture on the right side, and we see from that lesser curvature, and it does a good job here showing that lesser omentum binding up and behind that liver. Now, while we're looking at the liver, we're going to look at one more structure. It is the falciform ligament. Now, one more thing that it doesn't show here is the round ligament, but we will see a picture later on. So I want to introduce exactly what this is. So we see it right here. It is a ligament that's going to attach the liver to the front body wall, and its main goal is going to be to separate the liver into left medial lobes and a left lateral lobe. Now, we talked about fetal shunts when we did cardiovascular. Or the cardiovascular system. We talked about that fossa ovalis and the ligamentum arteriosum. Now we have another one here, and this is a remnant of the umbilical cord. We see that at birth, the umbilical vein is going to persist and be maintained as this round ligament. If you think of this structure, the falciform ligament and the round ligament, as a sail of a sailboat, and we have the sail that comes down and the boom that holds the sail at the bottom. That boom is the round ligament, and the sail separating the lobes attaching to the posterior portion of the body is the falciform. And again, when we do the liver in depth, we're going to look at these structures again in a little more detail. Continuing with the peritoneum, we can lift up that greater omentum, so you notice we lift it up. And we see the mesentery. So we see the mesentery here surrounding and moving between the small intestine. If we were to pull this transverse colon down, behind it you'd see a swooping mesocolon. Since we completed our discussion on how to protect the organs of the gastrointestinal tract, now we're going to start at the top and work our way to the bottom. So we start with the mouth. We're going to look at all the structures of the mouth and see exactly what they do. The mouth is going to be formed by the cheeks, hard and soft palates, and the tongue. So if we look at the hard palate, we find that it's mostly bony, hence hard, and it's going to form most of the roof of the mouth. Now, if you feel this structure, use your tongue, push it to the top of your mouth. I want you to pay note to its shape. It is domed upwards. This doming is going to play a pivotal role when we manipulate food in our mouth with our tongue that we'll talk about in a little bit. The soft palate, which is outlined down here, is going to be mostly muscular. It's going to form the rest of the roof of the mouth, is going to support the uvula, which brings us to this punching bag, the uvula. The uvula bangs backwards and it covers the nasopharynx. It blocks the nasopharynx, preventing food from moving up into the nasal cavity when you swallow. On the side, we have our cheeks. Our cheeks are super important to help us push in on food, manipulating that bolus or that ball of food that's being formed in our mouth through the process of chewing or mastication. We have teeth. We're going to go through our teeth briefly, although you're not going to be required to know them, but I do like to review them because it is somewhat interesting. The average human has 32 teeth. Well, that's if you haven't had your molars removed or your wisdom teeth removed. That would give you 28. But for a full set of teeth, we have 32. The 3 pairs in back, 1, 2, 3, and on the same side, 1, 2, 3, and the same goes for our upper set, right? We're dealing with molars, flattened surfaces, broad. Their job is to grind food. If you think about a cow or a giraffe or a horse, and if you ever looked at the majority of their teeth, they use mostly molars. And the reason why is because they don't need tearing or cutting. Their diet is mostly plant-based, so they need to break through that cell wall and they need that grinding action. And if you take your mandible and you move it left and right, you kind of simulate that grinding action that a horse does when it chews. And if you ever watch a horse or giraffe chew, you'll see that their lower mandible moves big swings left and right, again allowing for those teeth to do that action grinding. We have our premolars, so 1, 2, 1, 2, and then the same up on top, that are going to allow for crushing and grinding. That brings us to our canines. Now, when you talk about animals in the world, we have one of the smaller canines found. They are for tearing food. If you go to cats or dogs who have larger canines, or even go back into the dinosaur era where we had Smilodon or saber-toothed tiger that had these massive canines whose job was to once that animal bites down into the neck of their prey, those canines will tear and cut into the carotid or jugular of their prey, allowing them to sanguinate. The next teeth we're going to talk about are our front 4. These are our incisors. These are going to be for gripping and cutting food so that we can manipulate it in back and chew and break it down. Now that we're finished with our teeth, we're going to look at some spaces. We have the oral vestibule, which is this space right through here. It is the space between the cheeks, the lips, the gums, and the teeth. Now when you pull this lip down, this lower lip down, we find a sail of tissue. We have a sail of tissue here, we have a sail of tissue here, and we have a sail of tissue here. Now I'm going to erase it just so we can talk about it, but I wanted to point them out. Now, what is this sail of tissue? They are known as frenulums. Now, these frenulums are important because they hold our external structures, our tongue, our upper and lower lips in place, but they have and can be manipulated. Sometimes you see people that get frenulum piercings, or if you see a clinical condition known as tongue-tied, some people are born with a larger lingual frenulum, we'll point that out in a second, which limits the ability for them to stick their tongue out, which gives them a hard time to make th sounds, etc. So we start with our inferior labial frenulum. It attaches the inferior lip to the gum. We have our lingual frenulum right here that's going to tie that tongue into place so we don't swallow our tongue. And then below that we have some openings. These openings are going to give us the ducts of the submandibular gland, where we're going to release secretions of saliva. And we'll get into the details of that in a little bit. As we move our way up, we see our tongue. It is lifted upward. It forms the floor of the mouth. Its job is to manipulate food for chewing and then push it back for swallowing. Remember, we're going to assign some more scientifically correct words to chewing and swallowing. We're going to assign mastication for chewing and deglutition for swallowing. As we continue up our list, we find a pair of tonsils. palatine tonsils, which is going to be between our arches. We have a palatopharyngeal arch, and we see that right here, which is going to form our fauces. This is the opening between the oral cavity and the oropharynx. Following that, we have our gingiva. If you go back to our AP1 bone labs, where we talked about the alveolar processes, those projections that outline our tooth socket, we now put a tissue on top of it known as our gums or gingiva. Above there, we have our superior labial frenulum. So if we have an inferior down here, we, by law, everything's balanced, is going to have a superior labial frenulum, which is going to attach the superior lip to our gum. So now we could talk about our salivary glands and exactly where they're located and what we do. We start with the parotid gland, and we're going to draw a line here, and we're going to say that it is the largest of our salivary glands. Its job is going to be to secrete salivary amylase, and the function of salivary amylase is to begin the breakdown of starches. Remember, our diet is going to be comprised primarily of complex carbohydrates and starches, so this is something that is very large and very complex. Remember, our body wants to absorb the most simplest forms as polysaccharides, polyglycerides. So we have to break down these starches, these sugars, these fats into its smallest component. And it makes sense to start it right here. Now, our parotid gland is going to be located on the sides of our cheeks. Now, interesting little digression. When we talk about disease and disorder, we talk about the viral infection that causes mumps. So what mumps causes is inflammation of our two parotid glands, again located on the lower portion or size of your cheeks. If you were to draw a circle from your ear towards your zygomatic bone down to your mandible, this would pretty much tell us where our parotid gland is. Now again, the clinical condition, people who suffered from mumps would get swollen parotid glands, and the doctors called them chipmunk cheeks because they looked like, if you've ever seen chipmunks store food in their cheeks, this is exactly what this virus did to those glands. Now, the submandibular gland is just that. It is located below the mandible. And the submandibular gland has two functions. It has mucus cells that's going to secrete mucus. So we'll see mucus secretions that's going to serve a function of lubricating that bolus of food, lubricating the bolus of food as it enters down the esophagus. The second constituent that it's going to receive is salivary amylase. Again, we're adding another enzyme that's going to help or start the breakdown of starches. That brings us to our sublingual gland. Now, the name tells us exactly where it is. It is located below the tongue, and it's going to secrete something called lingual lipase. So before we get into lingual lipase, I want to show you some consistency through our naming, our nomenclature. Look at our enzyme word, ACE, ACE, ACE. Enzymes that's going to help break something down. Well, when we talk about amyl, we're talking about starches. When we talk about lipase or lipids, we're talking about fat, triglycerides or glycerides. So remember, fat is a very large complex structure. Our body does not like absorbing it in this form that we take it in our body, in the form of triglycerides or polyglycerides. So we need to break it down. We need to break it down into a monoglyceride. Now, just like we did with monosaccharides, we have to convert this or start the digestion of this fat into the smallest component. So where else to start it but in the mouth? So we can summate our three salivary glands and say that they lie outside the mouth and empty their contents through ducts, which deliver saliva into the oral cavity. We outlined and talked about what they did, but we have to remember that the main function is to soften, moisten, and lubricate and start digestion of this food, the bolus, the particular circular bolus that's being formed with your cheeks, your tongue, and your teeth before you swallow it down the esophagus. We can look at pictures to show us exactly where the salivary glands are. If we look up here, we have our largest of our salivary glands, the parotid gland. We go underneath the mandible and we find our submandibular gland, and again under the tongue for our sublingual glands. And you can see the ducts coming out following to their secretions. The next structure we're going to look at is the tongue. Now, contrary to popular belief, everybody thinks the tongue is a muscle. It is not. It is actually 8 muscles. These 8 muscles are going to form the floor of the oral cavity. It is going to be composed of skeletal muscle. You can control this and manipulate this. You can try by sticking your tongue out, moving it to the left or to the right, and it's going to be covered with a mucous membrane. Its job is to participate in chewing, swallowing, and speech. So if you think about the function of our tongue, pay attention the next time you're chewing or swallowing or talking. As you chew, your tongue is going to help move food in and around that oral cavity. Your cheeks will push down back on, pushing it and rolling it to that dome-shaped hard palate that we talked about. Here's the best analogy that I've come up with. If you've ever made meatballs before, think about your hands. Your hands are cupped, dome-shaped structures that you use to make meatballs, to make them nice rounded balls. Your hands are never flat. So this mimics what's going on in our mouth. The tongue moving food left to right, the cheeks pushing it back in, rolling it up consistently again and again to the roof of the mouth, to the hard palate, forming that bolus. Now, the upper and lateral surfaces of the tongue are covered with papillae. We've talked about all the different taste buds found on the tongue, as well as our foliate papillae, which are or do not contain taste buds, but are there for food manipulation. Aside from our tongue, we have our teeth, and we don't go too much into the anatomy of the teeth, but I'm going to talk about it a little bit here because I find it interesting. The teeth project into the mouth and are adapted for pretty much mechanical digestion. That's their job, either chewing, tearing, puncturing, rupturing, grinding food. The outermost important part of our teeth is, well, at least for visible perspective, is the enamel. It is made up of calcium salts. Its job is to protect the tooth from wear and tear. You can damage this enamel by drinking too much coffee, by smoking. It discolors this enamel, it weakens the enamel, and as our teeth get yellow, aside from those stainings, as that enamel thins as we get older from use and abuse, whether it be grinding our teeth or just age, the yellowing that we see is because we start to push, or at least we can see the dentin through the enamel. So again, the yellowing of our teeth due to age, due to abuse, is in part due to, well, somewhat due to staining based on diet, but more importantly, the color of the dentin as we can see it through that weakened enamel. So that brings us to our dentin. It is calcified connective tissue. It's going to make up the majority of our tooth. We have a gingival sulcus, so this groove which our gum is going to invaginate in. We have a pulp cavity. That pulp cavity is going to be connective tissue that's going to contain nerves and blood vessels. If you've ever had a root canal, they will file away a portion of your tooth, leaving a smaller nub that either a post or a crown will affix to, and they will slowly drill this out. And as they drill out that pulp cavity, they destroy blood vessels, they destroy the nerve. This is why you usually get a mandibular block when you have, or that if you think back to that trigeminal nerve, when we talk about cranial nerves, you get a mandibular block that blocks all the nerves to the face, so you do not feel this drilling as it happens. We have cementum. It is a bone-like substance that attaches the root to the periodontal ligaments, which brings us to all of our periodontal ligaments. These ligaments, if you go back to this joint, it is a gomphosis, is a immovable synarthrotic joint. Those periodontal ligaments are going to anchor the tooth to the underlying bone. As we get older and we abuse our teeth, these periodontal ligaments wear out and wear away. There are other things we could do to destroy those periodontal ligaments. There are drugs that affect these ligaments, that weaken them, that make our teeth fall out, as well as other diseases. That brings us to the apical foramen. It is a hole, you see that bone term again, foramen, that's at the base of the root canal that's going to allow for blood vessels, lymphatic vessels, and nerves to enter that tooth. Mechanical and chemical digestion in the mouth. We've talked about digestion a little bit. We've talked about the parotid glands, the salivary glands, and the submandibular glands. We didn't really outline specifically what that type of digestion is called. So we deal with two processes. The first being mechanical digestion. This is going to utilize our teeth, the muscular contractions in our esophagus and stomach and small intestine. Mechanical digestion is the chewing that's going to help mix the food with saliva in our mouth, forming that bolus that's going to be easily swallowed. Now, in the mouth, we talk about chemical digestion, where that salivary amylase is going to convert polysaccharides to disaccharides. Notice, we're taking those starches, those sugars, and we're taking complex multi-chain sugars and converting them to a simple, well, not quite simple, but a two-chain sugar, a disaccharide. We also deal with lingual lipase here, breaking down some lipids. Chart 24.1 is a good summary of the digestive activities in the mouth, looking at structures like the cheeks, salivary glands, tongues, taste buds, and lingual glands, what they secrete, their activity, and their end result, putting a nice table into everything we've talked about so far. As we continue away from our mouth, down our alimentary canal, we hit our next important structure. It is the pharynx. As we've said before, it is a funnel-shaped tube that's going to extend from the internal nares to the esophagus posteriorly and to the larynx anteriorly. It's going to be composed of skeletal muscle and lined with a mucous membrane, again, lubricating and facilitating movement through this tube-like structure. Now, the big key here is going to be skeletal muscle, and we're going to see this weird modification continue through the esophagus as well. Now, if we think about skeletal muscle, a few things should come to mind. One, voluntary action, right? And two, that we can control this. That's what voluntary means. So it makes sense. Look at its location. It's a funnel-shaped tube that goes from pretty much the internal nares to the esophagus. Now, go back to our respiratory lecture. Our larynx is here. Remember that epiglottis that's covering the trachea? Think about swallowing. Think about muscular contractions, right? Smooth muscle is slow. It's deliberate, right? It's slow and steady wins the race. Where skeletal muscle is fast. It's like the hare. So if we put this to an old fable, the tortoise and the hare, skeletal muscle will be the hare, smooth muscle will be the tortoise. Now, if you think about this funnel, it is from mouth to about right where the larynx is, where the trachea starts, and hence that epiglottis. So if you think about that muscle contraction, do you want slow and steady to win the race? The answer is no, because if you relied on smooth muscle contractions here, it could take minutes to hours to move food from your mouth to the esophagus, which is where you're breathing. And I don't think anybody can hold their breath that long. So it was smart to put skeletal muscle here. You could control this. Swallow hard. You are controlling the muscular contractions of this funnel. So if anything gets stuck, you can manipulate it by moving food past that little respiratory passageway, past that laryngopharynx, and down into the esophagus. It is a method of protection. Which brings us to our esophagus. Now, the esophagus is pretty unique. It is a collapsible muscular tube that lies behind the trachea, posterior to the trachea, and is going to connect the pharynx to the stomach. Now, a unique thing about the esophagus, it is broken up into three regions: a first third, a middle third, and a bottom third. Now, the first third is going to be primarily skeletal muscle. The middle third, or the second third, is going to be a hybridized skeletal to smooth muscle transition. And the latter, the bottom third, is going to be primarily smooth muscle. So we start thinking about what this transition is about. Why are we transitioning? We also transition epithelium as we move closer to the stomach, which we'll talk about in a little bit. So we'll continue with this. Let's look at a histological view of this. We start with the top, and look, we start naming, and you're going to do this in lab. We start naming layers. We have the mucosa, right? And we see that the mucosa is going to be made up of this non-keratinized stratified squamous epithelium. We have the lamina propria underneath it with the muscularis mucosae below it. From there, we hit our submucosa, our muscularis. So we have two layers, a circular layer and a longitudinal layer, followed by an adventitia. Notice we did not say serosa. So our esophagus lies outlined. That serous cavity is not surrounded by a serous membrane, so it does not have a serosa, but instead has an adventitia. Now, to look at this mucosa of the esophagus, and specifically this epithelium, we're going to see that it transitions about an inch or so before it makes its way to the stomach. That stratified squamous epithelium is going to switch over to columnar epithelium, and that columnar epithelium is going to be maintained pretty much through the entire alimentary canal until we get into our later portions. So we're going to see simple columnar through the later portions of the esophagus, stomach, all the portions of small intestine, the large intestines, etc. So we will elaborate this in a little bit. Deglutition. So we already assigned a word to this, meaning swallowing. So the tongue is going to shape that chewed and lubricated food, that bolus. As it moves it to the back of the mouth cavity, it will rise against the palate, closing the nasopharynx with that uvula to make sure food does not go back up there. And the epiglottis is going to cover the larynx in one fell swoop. Breathing will be interrupted as that bolus moves down the esophagus, following skeletal muscle contractions, peristaltic waves as we continue past the laryngopharynx, the pharyngeal stage, down and further down into the esophagus. We can follow this bolus using two types of muscular contractions. The circular muscles contract in these circular contractions, and then we get these longitudinal muscles giving us these peristaltic wave-like contractions. Now, we have two types of contractions going on here. The circular contraction is going to move behind the bolus, helping to propel that bolus down to the stomach. Those longitudinal muscles work in front of the bolus, and what this does is it helps elongate the tube. Now, for this elongation, I like to use the analogy of a clown. And some people will start saying, oh no, not clowns, I'm afraid of clowns. Well, not this clown. This clown is a balloon clown. And if you think about a clown that blows up balloons, and you think about what do they always do before they inflate that balloon? They pull on it, they stretch it. The stretching of that balloon before they inflate helps to facilitate inflation. The body utilizes a similar mechanism. Those longitudinal muscles that give you those wave-like contractions in front of the bolus help to elongate and facilitate movement of that bolus down the tube, just like that balloon clown. Now, as that bolus travels down, it hits a sphincter. This is the lower esophageal sphincter, also known as the cardiac sphincter. And the reason why it's called the cardiac sphincter, because the first region of the stomach is called the cardiac region. Now there's a reflex here. When that bolus hits that sphincter will open, allowing for that bolus to move into the stomach. Table 24.2 summarizes digestive activities in the pharynx and the esophagus. Everything that we have talked about in a nice, neat little table. That brings us to our next enlargement of this tube, known as the stomach. Notice it is a J-shaped enlargement. It is made up of 3 oblique, circular, longitudinal, 3 muscle layers, giving us a great opportunity to serve its function, which is essentially a washing machine stuck on spin cycle. Let's go to general anatomy and we'll work our way in. Where that esophagus touches that stomach, we have our first region, which is known as the cardia. The big bulbous portion is known as the fundus. The major portion of the stomach is the body. This J-like portion here is known as the pylorus. And we have our curvatures that we've outlined before. Our lesser curvature followed by our greater curvature. Now, when we talk about sphincters, those ring-like muscles that connect and seal off the stomach, we have our cardiac sphincter, also known as the lower esophageal sphincter, and we have our pyloric sphincter right here. That pyloric sphincter essentially will close and will open when we release this chyme mixture. So once digested in that stomach, that mixture will leak into the small intestine. Now, a couple more things to outline. Let's erase this mess that I've made here, and we'll talk about some more structures. Now, if we go back to the pylorus, we get a pyloric antrum. It is the space that separates the body from the pylorus, so right here, and we have a pyloric canal. The canal, again, is that chamber, that region that occupies the pylorus. Now, one more thing to look at, and we're going to see this again in reproduction. See these folds? All these folds of the stomach give us massive amount of surface area. This is why our stomach, we see here, this is why our stomach can enlarge massively bigger than what it is at rest, allowing for food to occupy and swell. These folds are in the mucosa, and they are called rugae. We can look at the internal anatomy of the stomach. So again, this is good because we can actually see these folds right here. We can see these rugae. We can see the cardia, the fundus, the body, all our curvatures, our pyloric antrum, and our pyloric canal. Our esophagus is superior, and our first portion of the small intestine, our duodenum, is inferior. Now that we have looked at the anatomy of the stomach, we can now examine its functions. So the job of the stomach is going to be to mix that saliva, that bolus of food, and gastric juices that have been produced by the cells of the stomach to form this liquidy chyme mixture. The stomach is going to serve as a reservoir. It is a bulk storage chamber that's going to store food before released into the small intestines. It could take anywhere from 4 to 5 hours for the stomach to completely empty. So when we say that it is a bulk reservoir chamber, that is exactly what it's doing. So you say, well, what is it doing for those 4 to 5 hours? Well, we're letting that stomach secrete its gastric juices. It's going to contain hydrochloric acid, pepsinogen, which we'll look at that conversion of pepsinogen to pepsin with hydrochloric acid, intrinsic factor, and gastric lipase. So we see all these enzymes are going to play a pivotal role in digestion, chemical breakdown of food. So the mixing, the churning that's going on in the stomach is more a mechanical form of digestion, whereas this enzymatic breakdown here is going to be chemical digestion. And what's the goal? We're breaking down these complex sugars, these complex starches, these complex fats into its simplest monoglyceride, monosaccharide, its simplest form. We're also going to see hormone secretion here. The hormone secreted is gastrin, and it's going to be secreted into the blood. Now, if we go back to gastrin, it is a peptide hormone, and it's going to help stimulate secretion of gastric acid by these parietal cells. And in the next slide, we'll look at all these different cells and what they do. And the parietal cells of the stomach are also going to aid in gastric motility. So we say, well, what's the overall aim of gastrin? Well, it's going to be to get the stomach moving. It's going to be to talk to the small intestine, the duodenum, and the pancreas to help secrete their products and get them moving as well as talking to those parietal cells. This slide walks us through the histology of the stomach, where we start right here, the basic layers. We start with the mucosa, and we remember that this is our epithelial layer. We have our lamina propria, this peachy colored layer here. And we have our muscularis mucosae, so our band of muscles right here. Now I want you to pay note to this lymphatic vessel that is moving through the stomach. This is important for removing various substances. These include cellular waste, water, and proteins from the interstitial fluid. As we continue down, we see our submucosa, that connective tissue with blood vessels, lymphatics. We have our muscularis, 2 layers, followed by our serosa. Now, notice our 2 layers of muscles, oblique and circular, and our longitudinal layer, our 3rd layer. Remember, we are in the stomach, we should see these 3 layers. We also get the enteric neurons, so that myenteric plexus and enteric neurons inside that muscularis. Gastric glands and cell types in the stomach. We start with the most predominant cell in the stomach, which is and are our surface mucus cells. They secrete mucus. Now, these mucus cells work in conjunction with our mucus neck cells to secrete more mucus. And then we say, why? Why do we have so many mucus cells? Think about what's going on in the stomach. We're secreting hydrochloric acid. We're secreting enzymes that break down proteins, that break down chemical bonds in tissues. Well, we do not want our stomach to digest itself. So as a result, we layer our stomach with a protective coating. The protective coating is mucus. That mucus is going to protect from the high acidity of the stomach, protecting it from essentially digesting itself. And now we can get into the meat and potatoes of the system. We can look at our parietal cells. Notice they are triangular shaped, almost like a trident. They are going to secrete hydrochloric acid and intrinsic factor. So we say, what's this intrinsic factor for? We talked about this when we talked about a condition known as pernicious anemia. We talked about it with the cardiovascular system and blood. So basically, we need intrinsic factor. It's needed for the absorption of vitamin B12, which is used in blood cell formation, or that erythropoiesis that we talked about before. Well, the second half of this is hydrochloric acid. It's going to be essential for killing the microbes in our food. It's also going to start the denaturation of the protein and serves a pivotal function in working with the secretions of the chief cells to convert pepsinogen into pepsin. So that brings us to our chief cells. We see that it secretes pepsinogen and gastric lipase. So what is pepsinogen? And we're going to go through this conversion in a little bit. But pepsinogen needs to be converted into its activated form. That's where hydrochloric acid comes in. Converts pepsinogen into pepsin. Once activated, it's going to serve its role to help break down proteins into peptides. That brings us to our gastric lipase. So we see that word lipase, we're thinking fat. Its job is going to be to split our triglycerides into fatty acids and start breaking them down into our simple fats known as monoglycerides. The last cell in this player of events, which are found deep into these gastric pits, are G cells. Now their name tells us what they do. They're secreting a hormone known as gastrin. Now here we go. We outlined gastrin a little bit, but let's outline it in depth. Its job is going to be to stimulate the parietal cells to secrete hydrochloric acid and talks to the chief cells to secrete that pepsinogen. It will also communicate with the lower esophageal sphincter to increase motility of the stomach, as well as relax the pyloric sphincter at the bottom of the stomach so that it can slowly leak this chyme material into the first portion of the small intestine, or the duodenum. So we look at secretion and regulation of hydrochloric acid in the stomach. We look at our parietal cell. We talk about the hormones and the neurotransmitters that are responsible for kicking this process off. We start with acetylcholine. So we have an ACH, acetylcholine receptor. We talk about gastrin binding to its receptor and histamine binding to its receptor, who's going to activate the cell to give us a net product of hydrochloride secretion into the lumen of the stomach from the apical surface. Now we can look at this in a little more detail. We look at cellular respiration. We start with water and carbon dioxide. We use our carbonic anhydrase to make our weak acid again. We're talking about carbonic acid. Now we know that it is a weak acid and it will dissociate relatively quickly to give us a hydrogen ion and a bicarbonate ion. Now we're going to use our bicarbonate chloride antiporters to move bicarbonate into the interstitium while bringing chloride in. Now, we can take the chloride route. Chloride is going to move through this parietal cell and utilize those chloride ion channels, those proteins embedded in the apical membrane, the apical surface, to push it into the lumen of the stomach. Now, on the other side of our trident, we have our hydrogen moving to that apical surface. We're going to use proton pumps, our hydrogen potassium ATPase proton pumps, to move potassium into the cell and kick hydrogen out. Now hydrogen and chloride are going to rapidly combine to make hydrochloric acid. This is going to contribute to the increase in acidity in the stomach. Table 24.3 goes through the summary of digestive activities in the stomach. It puts a nice little table together for everything we've talked about: structure, what's in the mucosa, what's in the muscularis, what's in the pyloric sphincter, what activity they are doing, whether it be secretion, absorption, mixing, or movement, and their net result. So please read through this table as it puts everything together in a nice, neat little package. Our first gland that lies inferior and posterior to the stomach is the pancreas. Now we kind of deviate from our main tube because we need to talk about some of our accessory digestive organs and what they do, because they do play a pivotal role in aiding in the digestion of structures like carbohydrates, proteins, fats, and nucleic acids. So the pancreas is a gland. Its location is posterior to the stomach. It is going to produce enzymes. Those enzymes includes carbohydrases, proteases, lipases, and nucleases. Now each of these enzymes will break down their respective counterparts. Carbohydrases break down carbohydrates, proteases, proteins, lipases, fat, nucleases, nucleic acid. It is also going to produce sodium bicarbonate. Remember, sodium bicarbonate is our body's best buffering system. That chyme liquid that is coming out of the stomach is very, very acidic. So it makes sense that this pancreas is located right within that first 8 inches of the duodenum, secreting this buffer that's going to help buffer that acidic chyme solution coming out of the stomach, protecting the integrity of the small intestine. Now remember, the pancreas was 99% exocrine. We talked about this when we did the endocrine system. We said that only 1% was endocrine. So we are outlining that 99%. Now, what is being secreted through ducts? Well, it's all those enzymes we just outlined. And it will empty its components into the duodenum via 2 ducts: our main pancreatic duct, which is known as the duct of Wirsung, and the accessory pancreatic duct, known as the duct of Santorini. We will look at a picture on the next slide to go through these parts. But one last thing to outline. So we see here is our duodenum, the first portion of our small intestine. We see our pancreas, our head, our body, and our tail. Okay, now one last thing to notice. One more structure that's moving into and through the pancreas and also connecting to the small intestine is this structure right here. It is the common bile duct. It is the duct that's leading from the gallbladder, and we'll see exactly what that does in a little bit. Let's put it all together. We can look at the pancreas, the liver, and the gallbladder. Let's start with the liver. As far as digestion is concerned, it is making bile. So bile is going to be produced in the liver. It's going to move through our right and left hepatic ducts, and it's going to be stored in the gallbladder. Now, movement of bile from the liver through the left and right ducts passes through the common hepatic duct into the cystic duct. Now, when signaled, when stimulated with Hormones like CCK, cholecystokinin, will stimulate the contraction of the gallbladder to move that bile through the cystic duct down through the common bile duct, at which point it will be stored in, and we go to this picture over here. So we follow bile flow through that common bile duct and stored in that ampulla of Vater, also known as the hepatopancreatic ampulla, before it is squirted through our major duodenal papilla. Remember, papilla means a projection or a little nipple-like structure that protrudes into the lumen of the small intestine, secreting that bile and the enzymes from the pancreas through the duct of Wirsung. One last thing to point out is we do have a sphincter here, a ring shape of muscles. It is called the hepatopancreatic sphincter. It is also known as the sphincter of Oddi. And this is what's going to regulate secretion in and out of the small intestine. Now, let's go back. Let's look at our pancreas in depth. We see, again, our head, our body, and our tail. So 99% of the structure is going to be secreting digestive enzymes. It moves through our main duct, our accessory duct. It'll enter into two nipples. Here's one, here's two. This one and this bottom portion is larger because it is combining the bile duct to the main pancreatic duct, so it's called the major duodenal papilla. The smaller one, well, if the larger is major, then you should be thinking the smaller one is minor, is what leads from the duct of Santorini. And then we see our first 8 inches of the small intestine, the duodenum, winding its way here. I want to point out the liver and some of those structures we talked about. We have our right lobe here, our left lobe here. Notice the diaphragm up above and our falciform ligament. And like we said, our analogy of our sailboat with the boom and the sail, we have that boom, that vestigial structure from the umbilical vein, that round ligament. We can now look at a dissected view of the pancreas, liver, and gallbladder. We can find a oblique cut of the liver right here, the falciform portion of the falciform ligament, the gallbladder snugged and hiding right underneath that right lobe. We see the cystic duct and common bile duct moving down into the pancreas, the main pancreatic duct, and the first portion of the small intestine. I do want you to pay note to the color. So let me erase my scribblings here and let's kind of zoom into what's going on. I want you to pay note to the color of the gallbladder and the color of all these ducts that are leading to the pancreas, as well as the coloration of the first portion of the small intestine. Notice that it is green. Now we can introduce some old terminology to explain why this solution is green. So there's 2 main pigments of bile: bilirubin, which is an orangey yellow color, and when it's oxidized form, biliveridin, which is green. And this biliveridin tends to stain upon exposure tissues in the body, which is why we see green colorations through these tissues. We can put that flow of bile to a chart so that it makes a little more sense. We look at the liver and the gallbladder. We know that the liver is making all of our bile. This is important because bile is an emulsifier. It's going to help emulsify fat. And for those of you who aren't familiar with this word, the best way to think about this is, well, it's a breakdown of fat from a larger component into smaller components. A good analogy is to take a Ziploc bag, fill it with water, put a cup of oil. That oil is in huge droplets. Take that bag and shake it vigorously. You then break up those big droplets of oil into these little droplets of fat. Those little droplets, what you just did was emulsify that fat, breaking it down from bigger component into smaller components. So we deal with the right hepatic duct and our left hepatic duct, carrying bile to the common hepatic duct from the liver, at which point we will hit the common bile duct. We're going to store via the cystic duct into the gallbladder. The gallbladder is going to store our bile until it is needed. Bile will then move again through the cystic duct into the common bile duct, at which point it will meet up with the duct of Wirsung, or the main pancreatic duct, hit that sphincter of Oddi until signaled to be secreted into the duodenum. Now we can look at the histology of the liver. We'll look at it from a diagrammatic perspective as well as a histological perspective. Before we get into it, let's look at its major functions. We're going to see that the liver's main job is going to be to filter the blood that's coming from the digestive tract before it gets passed to the rest of the body. We're also going to see that the liver is going to act as a detoxifier. It's going to help detoxify chemicals and metabolize drugs, which is why it's important that our liver is healthy. With a poor diet, high in fat and preservatives and poor food choices and alcohol consumption, our liver can tend to become fatty. As those fat deposits get gobbled up by phagocytes, it can leave scarring or cirrhosis on the liver, which can lead to dysfunction, which means the liver loses the ability to do what it's meant to do, making the majority of our plasma proteins, detoxifying chemicals, and metabolizing drugs, which is why if you are diagnosed with fatty liver disease, you see that you will not be able to take certain medications to treat ailments, which is very detrimental to your health. So, let's have a look at this structure. We see that the liver is composed of cells known as hepatocytes. Those hepatocytes make up our liver lobule. That is set up like a six-sided hexagon. Now, at the center, let's zoom on in, at the center of our lobule we have a central vein. Extending from the central vein we have our hepatic sinusoids. In between are all our hepatic cells known as hepatocytes. Now, at the corners of our spokes, we have three things. The three things are known as the triad. Here we have one, a bile duct. We have a hepatic artery, followed by a hepatic portal vein. So, again, central vein in the center, triad at the end. Our spokes, if you would think of this like a wheel, are going to be our hepatic sinusoids. Now, what's going on here? In that portal triad, the bile duct is going to be delivering bile, so bile is going to be created in the liver, delivered through those branches into the bile duct, which will be brought down to the gallbladder to be stored. Look at blood flow, the branch of the hepatic portal vein. Blood is moving in from venous as well as arterial and mixing, which is going to bring us to an important phenomenon. We will talk about hepatic portal circulation and why in our book it shows it as being purple. Here we take a look at the histology of the liver. We see our central vein up close, and we move out to our spoke, and we have the triad. We have a branch of the hepatic artery, our bile duct, and our hepatic portal vein. Now, from a lab perspective, what would you need to know? You would need to be able to identify three things. Number one, that this is the lobule of the liver. The cells are called hepatocytes. You can identify the central vein, and three, the triad. So three things: liver lobule with hepatocytes. So liver lobule with hepatocytes. 2, our central vein, and 3, our triad. So blood supply of the liver, and this is where it gets a little bit tricky. And again, we show hepatic portal system as being purple for a specific reason. So let's look at our two sources of blood. One, we get oxygenated blood from the hepatic artery, which means it is oxygenated and nutrient rich. We're also going to get deoxygenated blood from the hepatic portal vein. Now, typically in the body or in models, it'll show it as being purple. And the reason why is because it is traveling in veins, which means that the blood needs to be deoxygenated. So it's deoxygenated. However, contrary to most veins in our body, we are nutrient rich. So why is this carrying nutrients? Where did these nutrients come from? Well, it came from the small intestine. It did its job, it picked up those nutrients, and now bringing it to the liver via those hepatic sinusoids. Now those sinusoids are going to lead into the central vein, then to the hepatic vein, down to the inferior vena cava, into the right atrium of the heart to be reoxygenated, at which point it will loop back up and cycle through again. Now we can summate the functions of the liver and the gallbladder now that we have outlined them in depth, and see that the liver is going to be a major storage center for carbohydrates, lipids, and site of protein metabolism. Remember, again, that the majority of our plasma proteins are made here at the liver. We are processing drugs and hormones here. Remember, all our toxins are drug toxins and drug metabolites that are being sucked up from the small intestine, moved through that hepatic portal circulation to the liver for processing. We're also going to be excreting bilirubin. We're going to help synthesize bile salts. We're going to store bile. We're going to actively undergo phagocytosis. What we are removing here are protein complexes, some small particles, and cell debris that is moving through that portal circulation. We are also going to activate vitamin D, which will help to increase the amount of calcium the gut can absorb from all that food that you are ingesting.